My interest, my encounter with junk DNA or selfish DNA happened that during my PhD, I got interested in a gene which was uniquely induced with following certain treatment. And this was in 1969. At that time, it was not known that, okay, it was known that these, what we call polyethylene chromosome puffs, which are opened up chromatin, that they make RNA that makes protein. But nothing was known that which gene makes which RNA, which makes protein. And because I had accidentally come across an experimental situation where I could activate just one gene and inhibit all others. So I thought I should be able to do this quickly, identify this. Of course, from 1970, I kept on moving from one place to another and couldn't do this simple experiment. Lack of funds, lack of facilities to do all that. Finally, towards late 1970s, we could develop in this lab. Once I moved here to BHU, we fabricated equipment ourselves because we had no money to buy ready-made gel apparatus and gel drying apparatus. One of my students fabricated with his own hands, but much less cost. And then we found out that this gene doesn't make a protein.
Oh.
And that was the time, this 81. 82 we published this. And 1980 was this term selfish DNA, junk DNA came out. and so we were wondering, did we pick up a gene which is selfish or junk?
Right.
I never believed that. As a biologist, having faith in evolution, having faith in power of natural selection, I could never imagine that the cell will make millions of DNA
which
copies
are of no use.
Which are of no use because that's the energy currency you're spending meaninglessly. So I thought it must have a rule. Some function it must have. But the selfish DNA was what Crick and others suggest was that this DNA somehow gets into our system and it's selfish, it just maintains itself without doing anything to the host genome.
Correct.
Susumono junk DNA was that this DNA functioned earlier, but it is just lying unused because that's what the word junk means. There's something which was used in earlier times, but is
not used anymore.
Part of them may be true in certain ways, but then what biological systems do is they repurpose all these sequences. Some old sequences were lying. Today it may not have function, but these can be repurposed, and we know n number of examples where this has happened. Repurposing an existing gene, it has mutated, become functionless, and very shortly it acquires a new function. And that's how evolution happens.